you electrify the plant. And then when the insect feeds on the plant, and that insect happens to be wired up and in the system, when it feeds, it inserts its stylets into the plant, and this completes a circuit.
In this episode, I chat with Dr. Astri Wayadande, a professor from Oklahoma State University from a department previously known as the National Institute for Microbial Forensics and Food and Agricultural Biosecurity, also known as NIMFAB. According to ResearchGate, Dr. Wayadande has over 60 research items, over 890 citations, and almost 4,000 reads of her works. Dr. Wayadande is well known for her work in electropenetrography, where insects are used to complete a circuit to better understand some of their feeding behavior. How are you doing?
Hey, I'm great.
You're doing all right during all this stay at home and...
Well, I don't like it, and I'm going crazy like everybody else.
Yeah.
But I'm trying to look at on the bright side, if there is one. And basically it means that even though I'm working all the time, I get a little bit more rest, and I can choose, some downtime if I need it, because I'm usually at home. And so my garden is there. So I've been planting flowers and usually I don't have time to do that until after finals is over.
Yeah.
For the first time in, 25 years, I have a beautiful yard.
Dr. Astri Wayadande, you did your bachelor's in entomology first in the University of California in Davis, California, so UC Davis. And so you were already doing your undergraduate degree in entomology when you started. So you started in entomology kind of early. What kind of inspired you to study entomology?
I was a floundering pre-vet who didn't like it. And they were going to kick me out of school. And I was one of these kids who had to have an epiphany, or I was going to be out of there. I was really very fortunate because I ran into a friend of mine, and he was an entomology major, and I had never even heard of entomology. And he was, saying how much he really liked it. And so I decided, well, I'll just take a class. So I guess my junior year, I took an introductory class in entomology, and it was like, ba-bang! I was a zoo major at the time, and it was a huge major, one of those weed out things. And I was getting ready to have to do horrible things to little crabs, or big crabs, I guess. And I didn't want to do it. And when I took Harry Kaya's Intro to Entomology course, I fell in love with it. I said, oh my gosh, all the things I'm interested in doing, I don't feel so bad about doing it with insects. Turns out later there's issues and stuff, but I absolutely, I loved it. I was in a community of other undergraduates and a few graduate students. And it was the perfect environment for a student like me, because that was the beginning of my recovery and change in trajectory to something that led to academic and professional success. We certainly both work with brilliant people who've been brilliant their whole lives and careers. But then there's a subset of people like me who did not start out that way. And it was by luck, by chance, by serendipity that we run into a few key people who offered some words of encouragement, who said, hey, you can do this. And or things like, hey, look what I did. If I can do it, anybody can. And that's what I needed at that time. And it was, just a few words of encouragement, because UC Davis is a pretty big weed out school. And I was going to crash and burn.
Oh,
and I tell this story to my students because they come to me and they're upset. Oh, I'm not going to get into vet school or whatever. Like, you know what? That's okay. I mean, I don't want to poo-poo people's professional dreams, but there is is life outside of professional school. And if you don't make it, it could very well be because, wow, maybe something better is waiting for you. And you just don't think about those other choices because you're not faced with it. And I've seen it over and over again. Yeah, mediocre students. I was a mediocre student, and luckily I was given an opportunity to claw my way back and get up to that close to a 3.0 so that I could qualify to go to graduate school. I didn't quite make it. I had to get in on probation. But once I got in, it was like, hey, just get out of my way.
That was your opportunity to flourish right there.
Yes. Yeah. And so I'm always looking for students who maybe they're struggling, but they have that drive or that, special something about them. And you know what? You can do this. If it's something that is really, really important to you and it's driving you, who am I to hold you back because you're not a or even a B student? Some people are right on the cusp, and so okay, what do you need? And then get out of their way and watch them grow. It's amazing.
That's kind of really nice to hear, because I think for anyone who's considering graduate school, it is very different than undergraduate degree. I mean, like you said, I also came from one of those institutions that, especially those earlier year courses, 600 or 700 students. I mean, it's a weed out course, whereas as a graduate student, your advisor is trying to mentor you to become the best that you can, or at least to achieve your particular goals. So it's very different.
Very much so.
And so going now, so who were some of your inspirations then? So you mentioned, you had that switch. Who were some of your early inspirations?
Well, okay, so to get me into entomology, it was really serendipity. Dr. Harry Kaya was at UC Davis, and he was an nematologist. He's retired now. But he didn't usually teach the undergraduate, general entomology course. But anyway, so Harry taught that one quarter, and I was lucky to be in his class with a whole room full of like entomology wannabes. That these people became my peers for the next several years. So it was certainly Harry Kaya, because I learned so much from him. But then there was a graduate student that I worked in his lab. This was Dr. Jeff Garnett's lab, and there was this graduate student. His name was Tim Dennehy. And Tim is now like this big shot IPM guy. He's working for private companies now, but he was at University of Arizona and at Cornell for a while. But he could see that I was struggling, and I was his dishwasher. I washed his dishes. And he sat me down and he said, you think that you're here because UC is there to teach, but they're not. They're in the research business. And you have to figure out a way to set yourself apart and to survive and everything. And said, you can do this, you just have to persevere. And so if it wasn't for Tim Dennehy, I'm quite sure I would not be sitting here today, because he picked me up when I was really feeling low and said, yeah, you can do this. And so it's like, okay. And so, those two people, while I was at Davis. And then of course my two graduate advisors, Elaine Backus at University of Missouri. She was awesome. She taught me so much about research and about what I do now, EPG, and about behavior. And then my doctoral advisor, Skip Nault. What an amazing man. Ecologist, vector biologist, just all around inquisitive and exciting research scientist. That's exciting.
Wow.
I mean, I wanted to go to work every day.
That's a good feeling. So after your undergrad, so you already kind of mentioned some of your graduate advisors. So you did your master's in entomology, University of Missouri. And right after that, you went on to your PhD. What made you decide to go to your PhD?
I wanted to be just like Skip Nault. Yes.
All right.
I met him at a meeting oh gosh, it was back in 1986. And I was just like, oh my gosh. I mean, he must have been in his early 50s, and he was just so enthusiastic about leafhoppers and the pathogens that they transmitted. And I just like, I want to be like him. Do you ever have that where you see
Oh yeah.
someone? And so it became a singular obsession. I finished up my graduate work and I only applied to one school for my doctorate. Only one person I wanted to work with, and that was Skip Nault. And
Wow.
he
Yeah.
took me as a student, and yeah, it was rocky, and things didn't work out. But then a miracle happened, and then I ended up having the best project of all times. And
Oh,
so
wow.
it worked out really well. We're still
Yeah.
pals, today. So
Oh, nice.
yeah, it is.
What was the main, your main research for your PhD?
I was a vector entomologist in training. So I worked with leafhoppers that transmitted plant pathogenic bacteria. So mostly spiroplasmas, but there were a few viruses. So I had like a two type of system. I was working with feeding behavior of leafhopper vectors, and then also some really cool and interesting transmission biology of a semi-persistently transmitted corn virus, maize chlorotic dwarf virus. And
All
so I
right.
got to tease out some of the behavioral aspects of MCDV inoculation using electropenetration graph.
Wow. And
It
we're
was
and we're
cool.
going to get into some of these details of what some of this stuff means when we discuss the paper.
Sure.
But just in general, for a lot of people, some people listening might not be aware that plants can also have viruses and pathogens, and insects can
Mhm.
vector those viruses and pathogens, just like mosquitoes vector, viruses for us. And this can be very detrimental. That's where you have very critical issues when it comes to pest management, because it's one thing if you just have a certain percentage of leafhoppers or aphids or some other key pests in your crop, they can control with an insecticide. But if they are persistently or have been vectoring some kind of a virus, I mean, you can lose your entire crop. You could treat the aphids, but if your plants all have that virus, you might lose everything.
It's too late at that point.
Yeah, exactly. And so let's see. So now you are associate professor and interim director of the National Institute for Microbial Forensics and Food and Agricultural Biosecurity, also abbreviated as NIMFAB.
Well, I'll have to correct you. It's a little bit old information. So
Oh, okay.
is it okay if I update you?
Please. Yes, absolutely.
Okay, so I was promoted to full. So that was, yeah, woohoo!
Oh, congrats. Okay,
Finally.
nice.
You see all this gray hair? So yeah, so that happened. And then also I was interim director. We've hired a new director. This is Kitty Cardwell. She came to us from USDA. And very recently we had a name change, and we are now, instead of NIMFAB, we are now known as the IMBF, Institute for Microbial Forensics and Biosecurity. And so the mission is the same. Understanding relationships between plant pathogens, their vectors, understanding microbial forensics. And then of course we have a mission towards agricultural biosecurity, understanding all the key players, training for law enforcement, research support, advanced diagnostics, things like that. So the mission hasn't changed, but the name of the Institute has.
So what is microbial forensics?
Ooh, well, okay, so you know what forensics is. Texas
Yes.
A&M is famous for its forensics program. Well, in the traditional sense, forensics is trying to trace back or trying to understand by looking at the evidence who may have committed a crime and attribution for that perpetrator. So microbial forensics is just like that, except that instead of regular physical evidence that we use that might be admissible in a court of law, we're using microbial footprints, if you will, or traces So you know what a microbial profile is, right? We have lots of different microbes, and they're present in different quantities, different species. There might be something that's very rare, but very characteristic of a particular site or of a particular insect. Um, we have our own microbial footprint in our gut microbes, for example.
Mm-hmm.
And so by using microbial forensics, we can trace back a footprint of microbes that might be at the scene of a crime or at a particular, say for example, in a food safety event, where you're trying to figure out where a food safety microbe might have come from. We can trace back to a source, things like this. We use all this kind of evidence to help us to understand what might have happened, when it might have happened, and even perhaps how something might have happened. But it's very complicated. But they're learning more and more about bacteria, also about viruses and viral footprints. You know, we have microbiomes, we have viromes that have very characteristic constituents that make up those communities. So
is this methodology currently commonly practiced, or is it still in research phase?
I would say both. So the term microbial forensics was coined by a plant pathologist who was in the FBI, Bruce Bedoli. Although there are other people who contributed, I think Bruce was probably the first one to come up with the term. And he recognized that there's lots of bacteria in the world, and that we now are capable of detecting those bacteria. With the advent of sequencing, we can find out just tons of information about who is on my cell phone, for example, okay? Or on this pen, if it was left behind, as some evidence, you could actually do a swap, extract the DNA or RNA if you wanted to, and then figure out what microbes are there. I mean, this is a very simplistic explanation, but if you could match, you know, the stuff on this pen to, say, the microbes on my hand, you might be able to get a fingerprint match.
Yeah.
Like, oh, you've got 18 species, those same 18 species, including a very rare one that never occurs outside of the state of Ontario.
Yeah.
You have a match or something like that. But the thing is with microbial forensics, that the evidence has to be so stringent that it will hold up in a court of law. And that's really what we do. And that is to understand what might be happening and what evidence might look like, or what we have to do in terms of diagnostics and evidence collecting, so that it would hold up. It would be so stringent that it would hold up in a court of law. And that's the difference between, you know, just discovering what's, you know, on a tomato or on my cell phone, and then getting evidence that can withstand the strict set of guidelines required to either convict or to show that someone is innocent of a particular crime. It has to be really, really good and above question.
Yeah. Which when it comes to molecular work, it seems like is already very difficult as is. So now combine, you know, bacterial, like a profile or a signature and relative concentrations, and then make sure that, you know, you don't have a false positive or a false negative in all that. And
that's right. And so that's the research part of it, is to understand, you know, how microbes interact, you know, how they survive on surfaces, the diagnostics, you know, part of it, you know, presence or absence is different than the strict guidelines required for court, and so we do research, we understand interactions, we work towards developing useful tools and validation of those tools. That's another part of microbial forensics, is to validate the tools so that they can be used the same way over and over again, and that we have the same high level of identification or description.
Yeah.
So
now there's There's a number of people doing some very interesting and neat research in that department. Do you mind like highlighting some of the types of projects that are being done there?
Sure, I love to talk about my colleagues. We're a rather smallish department compared to Texas A&M, but small but mighty, I suppose. So, for example, the two other research scientists that I work with are Dr. Francisco Ochoa Corona. He is an advanced diagnostic specialist. He develops tools that we work with. And so he's doing research right now on rosette disease that is impacting your state as much as ours and around the nation. So he's trying to understand the relationship between the virus, understanding the plant virome, and understanding how movement is happening. He's working a little bit with the vector and doing some really great stuff with new tools, movable PCR, so you can diagnose things in the field, and developing new, he calls them user-friendly diagnostic tools. You know, you've done PCR before. It's like, you have to take your sample, go up into the lab. You have to have all these chemicals and everything. But what if you could do something really fast and easy that the grower could do, on his ranch or farm? And that's what he's interested in doing, is bringing advanced diagnostics, straight to the grower or the rancher or wherever the need is. So that'd
be pretty remarkable, because right now, a grower would have to submit a sample to a plant diagnostic lab. They need to process that. That can usually take 2 or 3 days for them to do and get back to the grower. But if the grower had something in hand, that can make a huge difference in turnaround, in treatment, so on and so forth.
Yeah, so he's working towards that, so it would make it a little bit easier. I mean, you stop and think about it. We've already placed a lot of useful tools in the hands of our growers and our ranchers. And so this is just sort of the next step, a handheld, like a tricorder. That'd be great.
Yeah, exactly.
So there's that bit of work. Then there's some very interesting food safety research done by Dr. Lee Ma. And one of her cool projects is coming up with, it's called cold plasma sterilization of surfaces. And so she's a food safety expert, and it's not enough just to understand the microbes. She wants to get rid of them and make it possible for processors or even home owners to have a quick sterilization technique to get rid of harmful bacteria. And so she's working on cold plasma applications of food safety.
Now, as a research scientist, as a graduate student mentor, what are some of the skills or life values that you think have been most important to get kind of where you are today?
Learn how to make your own tools. I'm a tool maker because I'm poor. Actually, I'm not, but I grew up in labs that didn't have a lot of money. I had to make my own tools. So I can make anything out of parafilm, glass pipettes, and tape. I'm kidding, but I've learned to get along. Is this a skill? Creativity, is that a skill?
Yeah, or resourcefulness.
Resourcefulness, I like that
Yeah.
better. That's a better word. So resourcefulness. The other thing that I think was really important, and this is an attribute, I guess, and that is insane curiosity. Like ridiculous. Like, how does that work? Or why does that happen? If you wake up in the middle of the night and you start thinking about, something that you're interested in, like, I wonder, why did that hopper, perform that activity and not this one? And it gets to bothering you, and I've got to figure it out. So then you design an experiment, you ask the question, you conduct the experiment, like, oh, okay, now I get it. And then you move on. I mean, I don't know. That's, for me, that was it. Writing, of course, is a skill, which I did not have. And I have fought very hard to become a better writer, but it was a skill and an ability that I just did not have, and it held me back for a long time. And that's why I teach writing intensiveness in one of my classes, is I want students to have that skill, or at least the confidence to recognize that, hey, I'm not very good at that. I need to know more about it. So resourcefulness, curiosity, writing skills. Oh, and the gift for gab.
Gift for gab. What is that?
The gift to talk, blah, blah, blah.
Oh, okay.
I don't need slides. I don't need a script. I don't need notes. I just need someone to tell me when to stop.
And that has certainly been helpful because you can communicate your ideas or have a discussion with people with ease.
Well, I think that these are qualities that even if you don't have them, I mean, I tell my students, you're not going to believe this, but I used to be an insanely and painfully shy person. And they look at me like, what? Are you kidding? And I say, no, I had to practice. And so I think that is a skill, and it's something that you can learn. And even if you don't feel like, oh, I'll never be able to stand up and give a talk without notes or whatever, well, I bet you can if you learn a few tricks and if you practice. And so that's one of the things I encourage my students to do, is to learn to feel comfortable speaking in public or just talking about their interests. And so I think it's important. Yeah. I don't know if that answered your question. It's probably wanted something more technical, but.
No, I think that's exactly it. And I think writing is one that I can relate to. I feel like I've improved a lot in my writing during my PhD, but still have a lot to learn. I still struggle and still find it very challenging. But it's certainly, I mean, I feel like once you figure that out, that's that one missing key I feel like I currently have. If I can figure that out, that's really going to help me out a lot.
Well, I'm waiting to figure it out too. And so it's a journey, Irfan, and you will find this out. I mean, there are some people who are naturally gifted writers, and I'm so happy for them, but I'm not one of them. So it's, I have to work at it and, make myself. And so I think it's okay to admit that, yeah, maybe you're lacking in something, but you can also make up for it and work towards, getting better. And if you do that, you usually will.
Yeah. You conduct research now that uses this EPG, and you've been using this for a little while, right? And that stands for Electropenetrography,
Electropenetrography.
Electropenetrography.
Yes. Yes.
Excellent. And could you explain that in brief? Kind of how does that work?
Yes, very simply. Okay, so electropenetrography, at least with plant feeding insects, this is the easiest system to explain with. You have a plant, okay? And you have an insect that's feeding on the plant. Usually these are piercing, sucking insects, okay? So if you electrify the plant by putting a small amount of current into the soil or into the plant itself, you electrify the plant. And then when the insect feeds on the plant, and that insect happens to be wired up and in the system, when it feeds, it inserts its stylets into the plant, and this completes a circuit, an electrical circuit. And so electricity can flow up through the plant and then across through the stylets, and then the insect then acts as a variable resistor. And so then the signal passes through the body of the insect and up through the wire and then back to the system, and where the information is recorded on a computer. And the display is a waveform.
Yeah, and it's pretty remarkable because depending on how that insect or where precisely that stylet is feeding, you can have slightly different resistance and a slightly different pattern as a result of the waveform.
Absolutely. So you can get information about things like where the insect is probing, where the stylet tip is, where the actual liquid is going up. You get information about what activity is happening inside the insect, so you can measure things like the sucking pump. You can measure streaming potentials within the food and the salivary canals. You can measure when the stylet tips over, for example, for an aphid, when it breaks into an epidermal cell, you can measure the break in the membrane potential of that cell. And that's a very, very important behavior for acquisition and inoculation of certain kinds of viruses.
Mm-hmm.
So it's that sensitive. So you get all this really cool information that is then very quantifiable. And so you can measure activities of a plant feeding insect. And then it turns out you can do the same thing with blood feeding insects.
Yeah, so that gets us right into this paper, right, which was published in the Journal of Medical Entomology in 2020, so this year, and entitled "Waveforms from Stylet Probing with a Mosquito Aedes aegypti Measured by AC/DC Electropenetrography," which I think I can pronounce reasonably now. Yeah. So why was this study kind of interesting, or why is this different than what's been done?
Well, traditionally, or in the past, this is not the first EPG study of mosquitoes. It has been done in the past, but using older systems that are not nearly as sensitive. Okay, so this particular study is unique because for the first time, it's using modern equipment that's capable of detecting more than the older equipment was capable of detecting. And so you're getting a lot more information other than inserting the stylets into, a mammal and then taking the stylets out and then just straight ingestion. We were able to discern things like movement of the stylets down to a blood vessel, movement into the blood vessel, and then ingestion. That was very simple. Yeah, appearance of blood is a pretty good indication there's ingestion going on.
So y'all did this on human hands, and I love this. Anyone doing mosquito work, I had a fellow graduate student back when I was doing my master's. We'd play squash together, and that was the time right after then he would say, I have to go feed my mosquitoes. He was all sweaty. He knew that he'd be nice and delicious for them, and he would just roll up his sleeve and stick his arm in his mosquito cage to maintain his mosquito colony. Yeah. I just found that crazy. And so y'all used human hands here as well. So are you putting a small current in the person's hand, or how does that work?
It was actually quite simple. We found out that if you hold the electrode in your hand, all you really need to do is have the mosquito complete the circuit. You don't feel any, in fact, you don't even have to apply any electricity. Our bodies generate enough electricity or current to be detectable by the system. Wow. And so you just have to be holding it. And when the mosquito pierces your skin, it completes the circuit. And then activities going on at the stylet hand interface, as well as pumping activities inside the mosquito, are picked up by the equipment.
And some of the types of behaviors you're able to capture or quantify included surface salivation, stylet penetration to the outer skin, penetration of deeper tissues and location of blood vessels, active ingestion with engorgement. I mean, that's pretty remarkable because visually you would not be able to see any of that stuff. But using this technique, you can know specifically what part of their feeding cycle they are in. And why is that? Why would that be helpful or interesting for future work?
Well, okay, for example, mosquitoes make really short bites, right? Evolutionarily, this is advantageous to them because they want to get in, suck, and get out before they get squished.
Swap.
So, of course, what happens during a mosquito bite, they can transmit, pathogens to whatever host they're feeding on. And that's what we're interested in disrupting. And then there's other things, people are very sensitive to mosquito bites. So anything that could impact shortening the bite or keeping them from biting, keeping them from accessing blood, anything like that would be helpful and useful. So actually you can see these things, but it requires a pretty hefty video equipment and like mice and things like that. And you can
the
see
ears of mice, I think y'all spoke about.
In fact, that was a wonderful paper that I used as inspiration, if you will. I wonder if you could capture this, but on EPG, which is a lot easier to do than to get a poor mouse, strap it down, anesthetize it, get the ear ready, get the mosquito in just the right spot, and then film. With EPG, you just wire up the mosquito, which is very easy to do. Get her on there, hook her up, give her your hand, and then if she's in the mood for a blood meal, boom, bada bing, bada boom, you're done in like 10, 15 minutes.
Wow.
And yeah, and then you can.
It's like donating blood. It's like donating blood. Incredible.
Well, yes, yeah, you could put it that way. But in terms of practical application, if you wanted to test things like, okay, I'm type A blood, all right, and I'm a mosquito magnet. I'm a self-disclosed. Mosquitoes like to bite me. What kind of person are you? Do they like you?
I think they like me a little bit less than my wife, but I don't know what I am.
Well, it may not have anything to do with blood type. So let's say that you don't attract many mosquitoes. What's the difference between you and me, other than, okay, you're a man and I'm a woman? Okay, there's probably other things. We have differences in our skin chemistries, our blood chemistry, whatever. There's a difference between people. There's variation in people. Some people actually will kill mosquitoes if they feed on them. There's something toxic about them.
Oh, wow.
So, trying to understand what these factors might be would be a wonderful application for EPG. Okay, you've got a mosquito, it bites you, but it doesn't take a full blood meal. What's going on? Is it have trouble finding the vessels? Is it constantly in that searching mode? Does it ever find? It can't. Okay, so at some point it's detected by the host and out it goes. So there are differences in people or animals, and if we understood what those were, we might be able to identify key factors that could be exploitable to develop better repellents or hey, have you ever wondered if you ate garlic, does that really help you repel mosquitoes? That's the next big experiment I want to try, is like massively, spaghetti with tons of garlic. And do I see a change in the way mosquitoes probe on my skin?
Yeah. I mean, if we're having massive meals, please let me know. I'll be a part of that. If it just means I got to put my hand in with one female mosquito, I'm in.
Wouldn't that be something, is if you detected something right, that just by consuming it was a regular food, and it caused just a change in your attractiveness to mosquitoes? I mean, that could be, something very useful.
Yeah, because I mean, they use not only like CO2 coming off of you, but also odorants, heat, and visual cues.
That's
so
right.
I mean, if you can get all of those on your side, then you can help repel them and reduce mosquito bites.
So, I mean, to me, those are the applications, is to try to understand how to thwart biting. If you can reduce or prevent, biting, or at least salivation when they're probing your skin, you could reduce transmission of mosquito-borne, pathogens. And ultimately, that's the goal.
Yeah, and that's, about 20% of all infectious human diseases worldwide, I'll quote, is from mosquitoes or mosquito-borne viruses. And that's huge, right? Mosquitoes are a huge, probably one of the biggest animal killers that we have on the earth. And yet we fear sharks and, alligators more. But, that's kind of one of the things, right, is, really knowing, being able to quantify this through this technique really helps with repellent or insecticide development, because if you can develop something that, even though a mosquito gets on you and will try to probe but can't quite get into that tissue, so it's transmitting that virus, that would still be considered perhaps a successful kind of repellent or a successful insecticide.
Yeah, what I'd like to do is, I've been working with insect vectors of plant pathogens for so long, and we really understand the acquisition and inoculation process. I don't think we understand it quite as well with vectors of human and animal pathogens, simply because it's really hard to take samples out of a living organism to study those interactions during the feeding process. You can do it with plants pretty easily, But with EPG, you might be able to, get a happy medium in there and to study some of these processes and understand a little bit more about how pathogens are inoculated, maybe how to prevent inoculation. If you could understand the precise moment when inoculation occurs within like seconds, there may be something useful that could be exploited. And I'd love to see other people learn this technique and exploit it for as much as we could get out of it.
Mm-hmm. Well, I thought this was a really neat paper. I want to thank you for doing such great research on mosquitoes and starting to tackle mosquito-borne viruses and how we can, at least develop some more techniques to better understand it so that we can tackle this global issue. And so thank you for that. And, I really appreciate your time with me today.
It's been fun. It's good to see you again. And I'm so glad that you're doing this. There's lots of people who are like stuck in their homes, or I'm all by myself in my little office. I'm practicing social distancing.
Yeah, I got mine here too. Yeah.
And I think it's really important to stay connected with your colleagues. And so
yeah, absolutely.
I do appreciate it.